So welcome back, class, to BIOM-MECH 543. This is Unit 6. You're almost done. You got 2-3 more units left, so keep on hanging in. You're doing great, I hope. There'll be 2 lectures in this unit. In Unit 5, we completed, we started talking about surface modification and functionalization methods, and that's where we left off. It's a very, very hot topic in the research of membranes. And so in this unit, we're going to complete our discussion of surface modification methods, talk about all the different functionalization methods there are, of which there are many, and why one would be better for one application than another. And in doing so, you're going to realize that this unit is going to be very chemistry heavy. So hopefully it doesn't scare you guys too much. Chemistry is the central science, right? Everything we do, biochemists, biologists, everything, it's all, if you boil it down to the fundamentals, it's chemistry. This is going to be fundamental chemistry, so just kind of bear with us on this one, but hopefully you'll see the application makes it worthwhile. We'll also start to talk about more niche applications of modification, where we can actually make these surfaces responsive to different stimuli. And that's advantageous in different ways. For anti-fouling properties, you can talk about changing temperature. There's a certain polymer that you can graft onto the surface that will swell and become very, very, like the polymer chain will become very long above a certain temperature, and it'll contract below a certain temperature. That can have some applications in drug elution and other things. You can talk about responsiveness to pH. Say if something's protonated or deprotonated. Usually when it's protonated, you end up with a more neutral charge that becomes more hydrophobic and such and such. And if it's deprotonated, so your pH goes up a little bit, it becomes more hydrophilic. So your membrane character basically flips. So that's kind of a neat new research method that we're talking about here. And so you can get changes in pore size, the porosity itself, the hydrophobicity, etc. We can also, with this functionalization, we can design multifunctional membranes. So not just separating based on size or charge, separating based on size and charge and hydrophobicity and affinity. Why not? We can mix them all together. And what this ends up doing is it lets us separate very, very complex mixtures and boil them down. Boil is not the right word, but separate them out into their constituent parts in a relatively low-cost manner compared to what would need to be for the other different applications. And finally, we'll start to move away from membranes just as separators. To date, I think we've talked almost exclusively about membranes as separating one thing from another. So obviously a big biomedical application is cell, tissue, and organ engineering. They can be used, membranes can be act as a scaffold. So cells being grown into organs, they need to be stimulated to secrete, the correct growth factors and the correct extracellular matrix. But they also need to have a correct geometrical orientation, and membranes can serve that purpose as the cells can stick to it. So you can basically mold the membrane to look like a piece of a liver or something like that. And that's actually a very, very, I personally really like that topic. And that's a very hot topic in research today. So we'll be talking and ending up the lecture with that.